biology students, today we're going to be talking about the cell cycle. Let's begin. The cell cycle is like the day in the life of a cell. Just like how you have everyday life stuff you do, the cell has everyday life stuff it does. But the end goal of the cell cycle is for the cell to go from being one cell to two identical cells. All right. The first part is called interphase, and actually in a second we're going to separate interphase into three separate chunks. But interphase, pictured here in blue, these three different parts, is the longest part of the cell cycle, and that's why we split it up into three different sections. And it's the busiest time, all right? And that's because that's when regular stuff happens. It would be the equivalent of your daytime, right? And it is when all of the cells' metabolic processes occur. And what does that mean? Metabolic processes means the chemical reaction. So all the things that enzymes do, cellular respiration, moving things around the cell, etc. Those are going to happen during interphase, regular functions. And that's why it takes so long. And it's pretty complicated, so we break it up into three major parts. Let's jump into those three major parts. The first major part, labeled number 1, is called G1, and the G stands for grow. The cell is mostly growing during this part of the cycle. So we go from being an original tiny little cell, and the cell eats, and it does its life stuff, and it gets bigger over time. Now we know that cells can't get too big, otherwise they can't transport things in and out of the cell membrane very well. But so eventually when it hits a certain size, it'll stop growing, but it can grow a little bit. That's pretty much all of G1. The cell just gets bigger. Let's do the next one. Number 2, S, synthesis. What is it synthesizing or making, which is what synthesis means? Well, we're synthesizing genetic DNA, genetic material, which means we are replicating it or copying it. So really, what are we doing? We're copying DNA here. So DNA at the beginning of the S phase will look like one butterfly wing, and then at the end it'll look like two butterfly wings for a full butterfly. And this is really important because at the end of the whole cell cycle, we want to go from having one full copy of the DNA to having a full copy of the DNA into two different cells. If I didn't replicate it, what would I do? I would have to split this guy in half, and then my two cells at the end would get partial amounts of DNA. No, what we want is that at the end, identical DNA in both of the end cells at the end of the whole cycle. So we have to copy that DNA here in S. Okay, so at the end of S, we have two copies. One is each side of the butterfly. Next is G2, which you would think would be growing, but yeah, it's some growing. But also we're double checking that we're ready to do cell division in a little bit, which is the end of the cell cycle. And to double check that we're really ready, the cell has a checkpoint at the end of G2. And this is like Gandalf saying, "Thou shalt not pass!" And it double checks the following. And if the following doesn't occur, then the cell really gets stuck here and can't move on. So what does it check for? Is the DNA really replicated or copied? Am I going to have full copies for two different cells later on? Do I have copies of all my organelles like the ER and the mitochondria to make enough for two new cells at the end? If I don't, what's going to happen? The problem is if I go into the division part, that's the shorter part, I'm going to now be kind of messed up, and this might lead to diseases down the line. So it's really great that the cell has this checkpoint. Sometimes, unfortunately, people's checkpoints here don't work that great, and we'll learn about that in a second. But hopefully we double check that we're ready to move on. So, summary, we had three parts of interphase. Interphase as a whole is very, very long, 70 to 90% of the cell's life or the cell cycle. Wow, it's almost the whole time. Let's jump into the next thing. So we're now done with those three stages. We're now ready to undergo cell division. And that is called mitosis or mitotic phase. That's what we call the M part. So this is much shorter Okay, and that's okay because all of this stuff happens pretty quickly usually for most types of cells. So we're not going to learn every step of mitosis right now. We'll do that at another time. But here's what the big important stuff that happens in mitosis. Really, the cell is dividing, all right, its nucleus. The cell nucleus gets duplicated. Those chromosomes that are copies, they're going to now get split evenly across two new cells so that we have two identical new nuclei. So this nucleus should have the same exact DNA as this nucleus. If for some reason this guy is slightly different than that one, we have a problem. So this is the division of the DNA and the nucleus. That's what mitosis is. At the very end of mitosis, there's a weird process called cytokinesis, which is referred to as cyto-cell kinesis. Think about telekinetic, which is moving things, cell movement. Now, after I've divided the nucleus in mitosis, I still have to split up my cytoplasm. Cytokinesis is somehow able to karate chop my cytoplasm in half so that I do get, in the end, two split up cytoplasm and split up cell membrane. Without cytokinesis, I kind of have these like conjoined weird twin situation. So thank goodness for cytokinesis. So in summary, just make sure you understand that all these things happened for a reason. At the end of the cell cycle, I have now two identical, genetically identical, meaning the DNA is the same, daughter cells. Meaning there was an original parent cell and now I get two daughter cells that are the exact same amount. Okay, so for humans, my normal number of chromosomes or DNA strands is 46. So at the beginning of the cell cycle during S, I copy them. 46 plus 46 is 92. And I needed to double them initially because at the end I need to split them. So again, I get 46. So I have a normal human at the end. So you have 46 chromosomes, I have 46 chromosomes. This process makes sure that in you, as you accidentally get a paper cut, the new cells that form are going to be identical to the old cells, genetically the same. Thank goodness for the cell cycle. Unfortunately, sometimes things go wrong, and this genetically identical stuff gets messed up. How does it go wrong? Well, we call that a tumor. Somehow, because of some environmental damage, there has been something that has affected my DNA. Maybe a mutation, which we'll learn about later in the year. And that DNA might not send that "thou shalt not pass" signal. And we might end up having a mass of cells, which means a bunch of cells in a space, a lot of cells. And we can have a benign tumor, which is an abnormal big clump of cells, but they're normal or healthy cells. So this is not a bad tumor. This is actually an okay tumor. Or we can have a malignant tumor, which is abnormal in size, but actually cancerous. So somehow our normal cell division, something went wrong in the process where we normally were going through the cycle of interphase, mitosis, cytokinesis. Somehow it got spun out of control and we end up getting a lot of bad cells. And that's not good. And it can tend to make someone sick if they're continuing to grow out of control, and if they accidentally are stealing nutrients and making toxins that make the person sick. In class, we're going to learn a lot more about cancer, but this is just the start. We get even more nervous if the cancer starts to move. This is called metastasis or metastasis, depending on who you're asking. This is when our not healthy cells that have some kind of miscommunication in the cell cycle, they divide out of control, but on top of that, they have started to get to a blood vessel and start to travel to other parts of the body. This is not good. So when we think about how bad types of cancer can be, we get most nervous not only if they're malignant, but also if they're moving, which is called metastasis. In class, we're going to talk about some ways to avoid different types of cancer, as well as ways to try to solve certain types of cancer and why finding cures to cancer is so difficult. Great job, guys. See you in class.